lecture, we're going to discuss the various types of genetic mutations. Following this lecture, you should be able to compare and contrast the three types of Mendelian inheritance. List and define the types of alterations that can occur in DNA, such as polymorphisms, mutations, and chromosomal aberrations. Identify the various mutation effects. List the environmental mutagens and describe the mechanisms they use to alter DNA. List and define the various types of mutations and be able to identify each regarding their effects on DNA. These include conservative and nonconservative mutations, point mutations such as missense, silent, and nonsense, frameshift mutations, deletions, insertions, and inversions. You should also describe the effects that various mutations have on gene expression and protein formation, and describe diseases that are the result of DNA damage and mutation, such as xeroderma pigmentosum and ataxia telangiectasia. So we're going to review the Mendelian inheritance patterns, and in an autosomal recessive disorder you need 2 copies in order to see the effect. So both parents must pass on that gene. In an autosomal dominant disorder, only one parent has to pass on the altered gene in order for that trait to be expressed. In X-linked disorder, this is a little bit different because usually everyone has 2 copies of each gene, and that's not true with the male sex chromosomes. So females, that is true because females have 2 X chromosomes, one from the mother and one from the father. However, males inherit 1 Y chromosome from the father and 1 X chromosome from the mother. The X and Y chromosomes both contain many genes that the other does not. And because males only have 1 X chromosome, they only have 1 set of genes that reside on that chromosome. So men and women differ in how they inherit sex-linked diseases. So in females, if a woman inherits 1 copy of a recessive gene on an X chromosome, she may not develop that trait because she has a normal copy of the gene on the other X chromosome. A female would have to inherit that recessive gene from both parents in order to be affected. In males, if a male inherits 1 recessive copy of a gene on his X chromosome, he will develop that trait because he doesn't have any other copies of that gene. He doesn't have another X chromosome. So men are more likely than women to get disorders associated with the X chromosome. So this schematic shows what happens with X-linked diseases. And women, if need 2 mutations, one from the father, one from the mother, in order to get the disease. However, in men, they only have 1 X chromosome, so if there's an alteration on that chromosome, they may get disease. So what are the different types of mutations that can occur in DNA? Well, one is a polymorphism, and a polymorphism is a genetic alteration that's harmless, and also it's present in more than 1% of the population, and that is what differentiates a polymorphism from a mutation. So poly means many, morph forms, so polymorphism. Polymorphisms may generate variations in traits or phenotypes. Some variations may lead to disease, and those that actually lead to disease are called mutations. So an alteration that may lead to a disorder, you can have a mutation, which is a harmful alteration in the nucleotide sequence of a gene, or a chromosomal aberration, where you have either an absence of a chromosome or absence of a large portion of a chromosome, or one or more extra chromosomes. And there are two different types of mutations. There are loss of function mutations and gain of function mutations. In loss of function mutations, you have an absent or dysfunctional protein, and this is more common of the two different categories of mutations. In a gain of function mutation, you have a protein or end up with a protein that has an enhanced function, and this is much less common. The effects of mutations. So once you have a gene that's been damaged or altered, that messenger RNA that's transcribed from the gene is also going to carry that same alteration that the parental DNA has. So the polypeptide made once that messenger RNA is translated may have a different sequence of amino acids. So the function of the protein may be changed, or it may actually be lost, because remember, proteins are linear folded amino acids, and if they don't fold properly, they may not be able to function. So the phenotype of an organism carrying the mutation might be changed depending on what type of mutation there is. Germline mutations are mutations in the germline cells, and these can be passed to subsequent generations. Once a child inherits a germline mutation, every cell is going to have that mutation. So germline mutations are responsible for hereditary diseases. A mutagen is an agent or a substance that can cause a permanent alteration in the DNA. So it's a chemical or substance that's going to change the DNA. So you have chemical mutagens, you have radiation, you have sunlight, you have spontaneous mutations, and all of these can cause alterations. So you have your DNA, your double-stranded DNA, and your DNA encounters a mutagen, and what happens is the DNA, the nucleotide sequence, gets altered. Chemical mutagens, there's many different chemicals that can damage DNA. So there are different mechanisms that these chemical mutagens can have. They might mimic a correct nucleotide base in the DNA molecule. They might remove parts of the nucleotide, causing improper base pairing during DNA replication. They might add hydrocarbon groups to nucleotides, and which is also going to cause incorrect base pairing. Radiation generates free radicals, and free radicals are highly reactive and will attack DNA and alter it in numerous ways. Radiation can also cause double-strand breaks in the DNA molecule. Sunlight contains ultraviolet radiation. So when UV light is absorbed by DNA, it causes a cross-link between adjacent bases. So usually our cells can repair this type of damage. Sometimes though, dimers develop and we don't catch them, our repair mechanisms don't catch them, and this leaves a gap. So sunlight can cause mutations in our DNA, and these mutations can be generated, and these can ultimately lead to things such as skin cancer and skin tumors if your skin is exposed to too much ultraviolet radiation. Exposure to ultraviolet light causes adjacent thymines to link, which is going to distort the DNA and break the hydrogen bonds between the thymine and the adenine. This schematic shows modification of nucleotide bases, where you have your double-stranded DNA and one of your nucleotides gets damaged and then a mutation is generated. So you can see on the left-hand side you have the normal sequence, and on the right the C has been altered to a T. And now you have a mismatch between the T and the G. Also, environmental agents can break the phosphate backbone of DNA, and this is what causes your double strand breaks to happen. Spontaneous mutations are mutations that occur without exposure to a known mutagenic agent. So sometimes DNA nucleotides just shift without any warning, and this is known as an isomer. So an isomer will form a different series of hydrogen bonds with its partner, which is going to lead to mistakes at the time that DNA is replicated. There are many types of mutations, regardless of what causes the mutation, whether it's an environmental agent, UV light, chemicals. The various mutations include both conservative and non-conservative mutations, point mutations, which include missense mutations, silent mutations, and nonsense mutations, frameshift mutations, deletions, insertions, inversion, and DNA expression mutations, and we're going to go through each of these. A conservative mutation is a mutation that might change the amino acid sequence. However, the new amino acid that's made has a similar biochemical property as the original amino acid, so the function may not be significantly affected. In a non-conservative mutation, you get a mutation that generates a amino acid that's biochemically different from the original amino acid, which is going to change the biochemical nature of the protein. Point mutations are an alteration of one nucleotide base. So in this example, you have an A that is altered to a C. So you're going to have a T on one strand matched up with a C. A missense mutation is when you have a single nucleotide that's altered, which causes a substitution of a different amino acid. So for example, on the top you have a GAA, and then your A is mutated to a C, and GAA and GAC are codons for two different amino acids. In a silent mutation, you get a nucleotide change. However, the amino acid is the same. So in this example, you have a GAA. The A is mutated to a G, and GAA and GAG are codons that encode for the same amino acid. A nonsense mutation is a point mutation that causes a premature stop codon. So remember, your stop codons like TAA, TAG, this is going to lead to a truncated and possibly a non-functional protein because that stop codon is too soon in the sequence.
In a frameshift mutation, you have one or more bases that are either inserted or deleted. So when you have an alteration in your messenger RNA sequence, that's going to be translated so you can get an alteration in your protein. So in the schematic, you have on the top ATG, GAA, GCA, CGT. On the bottom, you have your ATG, which is your methionine, your start codon. The G is deleted, so you have a one nucleotide deletion. Well, your triplet codon is going to start at the A. So instead of having GAA, your next amino acid is going to be encoded by AAG. So instead of having a glutamic acid, you're going to have a lysine. And then all of the subsequent amino acids might change from that point on because the triplet codon is going to be altered by one frame because you have one nucleotide that's missing.
A deletion mutation is a mutation that results in missing DNA. So these mutations can be very small, just deleting one nucleotide, or they can be larger, where numerous nucleotides can be deleted. Deletions can also cause frameshift mutations. So deletion mutations, it depends on what's deleted, where it's deleted, how much is deleted, if there's going to be an effect or not. Insertion mutations cause an addition of DNA. Insertions can also cause frameshift mutations because if you add a nucleotide in, all of your triplet codons at that point are going to change. So again, it can result in a non-functional protein. In inversions, you can have an entire section of DNA that's reversed. You might have a small inversion where you only have a few bases that are reversed. However, you could also have a very long inversion involving a large region of a chromosome containing several genes, and that can alter the protein. DNA expression mutations are a type of mutation that does not change the protein, but it changes where and how much of the protein that is made. So it could result in proteins being made at the wrong time, or proteins being made in the wrong cell type, or either too much or too little protein being made. So you can get mutations in regulatory regions and promoters, and that is going to affect how much that gene is expressed, which is then going to affect how much protein is made. And remember, the proteins are the workhorses of the cell. And if you don't have enough protein made for a particular situation, that might be detrimental to the cell. Some mutations result in proteins that don't function normally, and that could end up causing a disease. So there are different ways that a mutation can change the way a protein functions. They can cause altered protein function. They can completely cause that protein not to be made at all, so you don't get any protein, or alter how that protein is made. Some mutations cause a protein that won't be able to carry out its normal function in the cell, or not be able to carry out its function very well. And an example of a mutation that causes an altered protein function is the mutation that generates sickle cell anemia. So in sickle cell anemia, you get an altered protein in the red blood cells that alters the shape of those red blood cells. Instead of them being round, they become sickle shaped, and the red blood cells get stuck inside the blood vessels. also those cells are not able to carry sufficient oxygen to the rest of the body. You can have mutations that do not make protein at all, so prevent protein from being made completely. And an example of a mutation that causes a lack of protein is hemophilia. Hemophilia is caused by a mutation that causes an absence of protein that's needed for blood to clot. So it results in uncontrolled bleeding in response to any injuries. You can have mutations that change the amount of protein, either causing too much or too little of a protein to be made. So the protein will function properly, but its quantities are not sufficient. So an example of this would be several different types of cancers. So you may need a protein that prevents additional mutations from building up, and that protein is not being made at a high enough level. So without that protein, the cell is going to accumulate mutations, and that's where you get your cancer. Now, since we do have various types of mutations, our cells have DNA repair mechanisms, numerous types of repair mechanisms. However, these repair mechanisms don't always work, or they are work for some types of mutations but not others, or your body just skips a couple and lets a few mutations get through. So that's why mutations are always going to occur. But we do have mutation repair mechanisms, and if our cells didn't have mutation repair mechanisms, we most likely wouldn't live very long, because we would generate so many mutations that over the generations we would just get too many mutations for the cells to function properly. So there's many different DNA repair mechanisms. You have base excision repair, mismatch repair, recombination repair, nucleotide excision repair. We're not going to talk about all of these in detail, but just knowing that our body does have ways to prepare damage, DNA damage, and mutations that arise along the way. So DNA damage in our cells is very, very common. So we do have these repair mechanisms. So we have damage reversal repair, and these include photoreactivation and ligation of single-strand breaks. We have damage removal, which include base excision repair, mismatch repair, and nucleotide excision repair. And we also have damage tolerance, which includes recombination repair and mutagenic repair. So remember, we talked about the enzyme ligase, and when we have recombination in your lagging strand, you generate your Okazaki fragments, and then your ligase is the enzyme that's going to come in and glue all those fragments together. Well, ligase is also needed to repair single-strand breaks. So X-rays and other chemicals like peroxide cause breaks in the backbone of DNA. So ligase is going to come in just like it does in recombination with Okazaki fragments. It's going to come in and glue those breaks back together. We also have nucleotide excision repair. So you have DNA damage that can be very bulky, and it could block DNA replication and transcription. So if you get ultraviolet induced dimers caused by chemicals and things like that, what happens is you have a repair mechanism that's going to cleave that DNA strand where the damage is using an enzyme called endonuclease. Exonuclease is going to come in, remove the segment that contains the damage, and then your DNA polymerase is going to come in and fill in that gap with the nucleotides that are going to be complementary. However, we do have nucleotide excision repair mutants. So in these mutants in lower organisms, they're highly sensitive to ultraviolet light. What happens in these mutants is they have high levels of mutation and recombination that can be induced when you expose them to UV light. There's also an example of a nucleotide excision repair defect that causes a human disease, which is called xeroderma pigmentosum. Xeroderma pigmentosum is a genetic disorder where your repair mechanism doesn't work properly. So this is a hereditary disease, and these individuals are sensitive to sunlight. So if these individuals are exposed to sunlight, they have a very high risk of skin cancer. So these individuals have defects in genes that are homologous to those that are required for nucleotide excision repair. And these people cannot expose their skin at all. So in the middle of the summer, they have to wear big hats and cover every exposed surface of their skin. There's another DNA repair disorder called ataxia telangiectasia, and this is an autosomal recessive disease, and it causes numerous types of defects that increase cancer susceptibility. So in AT patients, in cell culture, if you take AT patients, take their cells and grow them up in a petri dish, you can see spontaneous as well as radiation-induced chromosomal breaks, as well as sensitivity to X-ray exposure. So these patients are extremely sensitive to X-ray doses, so they cannot be exposed to X-ray if they were to develop a cancer. The AT culture cells don't show a defect in the repair of X-ray damage. So unlike their normal cells, they continue to replicate their DNA even when it's been damaged by X-rays. So what AT does is it doesn't recognize that DNA damage. It's not going to respond appropriately and halt that cell cycle so that repair can occur. So what happens is you get chromosomal alterations and death if the AT patient is exposed to X-ray. So the ataxia telangiectasia is a defect in a cell cycle checkpoint. So this is where your cell decides to go on to the next phase of the cell cycle. So mutations in cell cycle checkpoint genes can lead to uncontrolled cell growth, which would lead to cancer. So AT is a very rare condition. It's estimated that the frequency of heterozygotes with one AT mutation is only 1% in the population. So these individuals also, in addition to being highly sensitive to X-ray, they have a high risk of cancer when they're exposed to any type of radiation. So they cannot get any type of X-ray procedure. So in summary, the things that you want to know from this lecture are: what are the differences between autosomal dominant, recessive, and X-linked disorders? What are the various types of DNA mutations that can occur? What are the effects of these mutations? What types of repair mechanisms do our cells have? And what types of diseases occur when these repair mechanisms don't work properly?